where every cell tells a story. I am Farid, your own embryologist.
So hello everyone. In this video now we will look at the cell counter. As we have already discussed about the Makler Chamber, so here I have also put it down. So yes, in the last video we have discussed about the Makler Chamber and we observed the working of Makler Chamber, not exactly, but we just take a look at the Makler Chamber, how it looks like and how you have to arrange it into the microscope, the way Makler Chamber is showing the 100 grids, and with the naked eye you won't be able to see a single dot, but when you put into the microscope, even in the 10x you are able to view the 100 grids, but it will be more specifically visible in 10x and 20x and 40x and 100x. So basically we have already observed it in the 10x. Now what? Okay, now you are it's like 100 grids are there, but you are considering the 20 grids and you are observing the 20 grids. So how you will remember that how much cells you have observed? You cannot count the cells, but what you will do, you will definitely go for the sperm cell counter. So basically what happened when you talk about the counting of the grids, so for the counting you need a sperm cell counter, which is also known as the blood cell counter. So this is the sperm cell or blood cell counter. So basically it will not only observe the count, it will also be helpful for the counting of motility. So basically as we were discussing about the count only, so I will keep it into the around the count only. So what we will do, we will pour the 10 microliter sample into the Makler chamber, into the center point of Makler chamber. Then we will align the cover slip in such a way that the two dots will be parallel, that we have already discussed. After the completion of this entire process, what we will do, we will put the Makler chamber into the microscope, and always remember while focusing, because focus is very crucial in embryology and andrology both. So while focusing you always have to set into the minimized lens, whether it's the 10x, whether it's the 20x. So in this case we are having the 10x, which is the minimized lens. So you have to put into the minimized lens, and at the time of focusing you have to be very careful, because as I have already told you that it costs 30,000. So it's not only about the Makler chamber cost, but it also about the lens cost, because if you put so much pressure and the lens or Makler chamber gets broke out due to any reason, then you will be find yourself into a lot of trouble. So basically what you have to do, you don't need to be fair about that, but you have to be careful. So just do with fine hands, just do like you are loving someone, the way you have to do very gentle touch or very smooth touch. You don't need to be rough, you don't need to be fast that okay I have to focus. No, just wait, just observe that okay, at one time you will get to know that okay, gradually these things are coming into the focus. So you have to give time and patience, and after focusing onto the minimized lens like 10x, you will shift the lens into the 20, or you can also keep it into the 10, it's up to you. And after shifting the lens into the next or keeping it into the same, you will start counting the cells like you are showing, like it's as it's visible into the screen, like you are seeing the 10 grids. Basically what happened, 10 grids specify the exact count, like you are seeing the collectively 10 grids count. So you have observed 60 cells. So 60 cells is the exact value. So the count will be 60 million. But for a safe side, we always keep two rows like 10 and 10, 20. So 20 rows will be taken, and these 20 rows will be considered as the collective value of the sperm count, and then we will divide it with 2, so that the average will be more accurate. So now, like you have observed like it's okay, you are looking into the lens and it's like you are looking into the microscope, and each grids are having different number of cells. But obviously you are loading a sample and that sample will uniformly be mixed, but the cells will not be uniform na. One grid will show 10, another grid will show 5, next grid will show 15, another grid will show 0. So it's like you have to keep counting the grids. So how you will count, it's like you will just keep it into the like this. Okay, wait, I will show you. So now like sample is loaded, the amount of sample I have discussed, how to focus we have discussed, what lens we need to prefer, we have discussed everything, we have discussed. Now let's suppose, first of all we will make it into the neutral. Now it's neutral. I will show you next time like this. It looks like this. It is the selfie camera, so you want to be able to see it in a proper way, but I will show you it about it. Now it's completely neutral. So how you will get to know that it's neutral? I will put a photo of that. Okay, so you just have to keep it into the left corner. This is the left. You will see into the microscope, and now both motility and count will be observed at the same point. But first of all, we will, as we are discussing about the count, so we will only discuss about the count. Okay, so for counting you can also pick one finger, obviously. So you have just put the finger into this and start looking into the microscope, like as we have discussed, and I said to you that we will keep counting the grids. So I have picked the one grid. Okay, so the one grid is having, like, it's not having the cells, but actually let's consider it's having the 3 cells. Okay, the next grid is having the 2 cells. Okay, the next grid is having no cells, and another grid is having 5 cells. Okay, another grid is having 4 cells. Another grid is having 2 cells. Another grid is having 3 cells. Another grid is having no cell. Another grid is having 2 cells. Now, how much we have counted? Let's wait.